The Red Queen hypothesis, also referred to as Red Queens, the Red Queen effect, Red Queen's race, Red Queen dynamics, is an evolutionary hypothesis, which proposes that species must constantly adapt, evolve, and proliferate in order to survive while pitted against ever-evolving opposing species. The Red Queen hypothesis is related to the coevolution of species. It states that species must continuously adapt and evolve to pass on genes to the next generation and also to keep from going extinct when other species within a symbiotic relationship are evolving. This refers to an idea in evolutionary theory, which states that all species are under constant pressure to evolve into better organisms in order to survive amidst competition. Rabbits, for example, need to continuously evolve to run faster over time, or their population would be completely destroyed by foxes. Foxes, on the other hand, will have to constantly evolve to run faster over time, or they would be unable to hunt down rabbits that satisfy their dietary needs for survival. Van Vallen coined the hypothesis Red Queen because under this interpretation, species have to run or evolve in order to stay in the same place, or else go extinct. The hypothesis was first proposed by American biologist Lee Van Vallen. The name is derived from a statement that the Red Queen made to Alice in Lewis Carroll's Through the Looking Glass in her explanation of the nature of Looking Glass Land. Now, here, you see, it takes all the running you can do to keep in the same place. Lee Van Vallen proposed the hypothesis as an explanatory tangent to explain the law of extinction, known as Van Vallen's law, which states that the probability of extinction does not depend on the lifetime of the species, or higher-ranked taxon, instead being constant over millions of years, for any given taxon. An example of the Red Queen hypothesis might be one of the plants that evolve toxins to kill off predators, such as caterpillars. If the plant, under predation selection pressure, evolved a new type of toxin, to which the caterpillar had no immunity, most of the caterpillars would die off, and the tree would flourish. This victory would be short-lived, as only the caterpillars immune to the toxin, even if only a tiny percentage, would breed rapidly, and once again the tree would be under attack. Another example is the random fluctuations and mutations of genes, to allow new traits to appear. The industrial melanism, shown by the peppered moth, is an excellent example of this process in action. Without this mutation, there would be a chance that the species may have become extinct. Industrial melanism refers to the genetic darkening of species in response to pollutants. Peppered moths are normally white with black speckles across the wings, giving it its name. This patterning makes it well camouflaged against lichen-covered tree trunks when it rests on them during the day. There is also a naturally occurring genetic mutation which causes some moths to have almost black wings. These black forms, called melanic, are not as well camouflaged on the lichen as normal peppered forms, and so they are more likely to be eaten by birds and other predators. This means that fewer black forms survive to breed, and so they are less common in the population than the paler peppered forms. Industrialization and domestic coal fires had caused sooty air pollution which had killed off lichens and blackened urban tree trunks and walls. So now, it was the pale form of the moth that was more obvious to predators, while the melanic form was better camouflaged and more likely to survive and produce offspring. As a result, over successive generations, the black moths came to outnumber the pale forms in our towns and cities. This also explains why the vast majority of genes in vertebrates are dormant and do nothing, junk genes, as they are preserving possible mutations that might suddenly be needed in the future if the environment or parasite pressure changes. One more example is sickle cell anemia. Malaria parasite cannot live in blood affected by the sickle cell disease. Only individuals with no copies of the gene are likely to suffer malaria, those with 2 copies anemia. The individuals with 1 copy of the gene will be unaffected, as they do not have enough cells affected to cause the anemic effects, but enough to deter the malaria parasite, the cost being that some of their offspring will suffer anemia or malaria susceptibility.